Yes, hi everyone. We have an update from NurExone Biologic Inc. It trades on the OTCQB under the symbol NRXBF and on the TSXV under the symbol NRX.
I want to give a small introduction and open it to a live dialogue in order to answer your questions. So NurExone is where nature ends, NurExone begins. We are talking on the hardcore transforming the regenerative medicine, how we do it. Not with the disclaimer. Disclaimer is there because we are a publicly traded company. Our mission is to develop regenerative medicine specialized in to recover tissue, damaged tissue of spinal cord injury and other viral indication of CNS, central nervous systems. Our first product called XOP10, okay, which target less than 250,000 people around the world. In the US, for example, is just 18K new spinal cord injury or acute spinal cord injury a year. And what we do is to regenerate, rewire the neurons, and recovery. And we do it in minimal invasive way. Just about the company, established in Israel, a tech transfer from the university, from the Technion and Tel Aviv University into our company. The model is hybrid model. We have an exotherapy platform and the product. And this exotherapy platform is based on 5 families of patents covering production of exosomes, loading of exosome post-production, and what we load into exosomes. And we are specialized in siRNA delivery system. Our target is to help patients with acute spinal cord injury and other traumatic CNS indication, such as traumatic brain injuries, optic nerve damage, etc. We are publicly traded company since 2022, so we have quite large history. You can follow us and see our activity in the last 2.5 years. There is a holding company and an R&D company 100% owned by NurExone Biologic Inc. Recent updates, we completed successfully the pre-IND with the FDA, and we are working now on the IND-enabled tests. And we have orphan drug designation in the US, and we are waiting, expecting to have a final results and resolution regarding orphan drug designation in Europe. And we are working intensively on the preclinical testing and development methods in order to get this product to the clinic in the next 12 to 18 months. I will skip the science. Exosomes are small vesicles. We use it as a guided drug delivery system from different reasons. One of the reasons, several advantages, we can use it in minimal invasive way. The drug delivery system has a therapeutic effect. We can use it off-the-shelf product. There is very low toxicity, if at all, of these products. So it's not a personal medicine. We can treat several patients with one batch. And really, it's really important and critical when you're talking about biologics. And it's crossed the brain-blood barrier, which exactly it's our target, the brain and the central nerve systems. And we can load post-production our drug. With that, I want just to highlight several recent activity that we had. We published in July results, not only in spinal cord injury, also in glaucoma model, optic nerve damage, very positive results in the early study that we had. We see a functional recovery of the optic nerve after a crash. And so this is the second indication showing the same product, regeneration, which gives you as the investors much more confidence in what we do. And we will continue exploring other indications like traumatic brain injury in order to prove concept that our drug can work in other indications. And with that, I would finish the short update and take questions.
All right, Lior, can you hear me?
Yes.
Okay, wonderful. Okay, Aiden asks if you can talk about recent studies and how far along are you in exopTN, if I'm saying that correctly.
Yeah, so exopTN, again, is exosome loaded with siRNA against PTEN. The first indication was acute spinal cord injury. We are the most advanced in this case. We are in an IND-enabling test, which is the tox study and preclinical development of the drug with the guidance of the FDA and the orphan drug designation from the FDA. And we have, as I mentioned, other indication, optic nerve damage, which is relevant for glaucoma models. We're using rat model with the compression of the optic nerve, treat with the same drug, exopTN, twice, once after injury and a week later. And we see 18 days after the injury, a normal ERG measurements. It is electrical measurements of the eye activity. It's called dark adoption. And we see that the normal eyes or the damaged eyes after treatment is behaving exactly like the normal eyes. Again, is the same rat. One eye is untouched and the other one was damaged and treated.
Wow, fascinating. We are out of time. We have to move on to our next presenter in just a few minutes, but we will send you the rest of these questions so you can answer on your own. But please join us again in the future with another update. Important work you're doing here.
I will come in next month on December. So great. Looking forward to be here again. Thank you.
All right. Thank you so much.
Bye-bye.
Stay with us. We'll be right back.